Metagenomics LoopSeq technology. So for those who have been involved in sequencing, there are sort of two main classes of sequencing technologies that we're more or less familiar with now. So first is the dominant short read sequencing platforms. Illumina is the big player there, although there are others. And they produce relatively short reads, hundreds of nucleotides, but they have good error profiles. They have low error rates, 0.1 to 0.5%, and probably closer to 0.1% for sort of modern Illumina instruments. The alternative sequencing technologies are long read sequencing. And so here are the two big tech providers are PacBio and Oxford Nanopore. And they produce reads that are much, much longer, 1 to 200 kilobases. And out of Oxford Nanopore, you can get at least some reads even much longer than that, although it's not really the bulk of the reads you get out. But they're also dramatically different in that the per base error rates of these long read technologies are 2 orders of magnitude higher than for short read sequencing. And once you're up into this range of sort of 3 to 15% error rates, that can really challenge some of the things that you'd like to be able to do with that kind of sequencing. So example, calling ORFs on Oxford Nanopore can be quite a challenging thing because all of these insertion deletion errors that scramble the reading frames. So this is just a little visualization. So the length of these reads is sort of shown to scale. On the bottom, we have this short read, and that's really what a length, I think that's a 200 nucleotide short read. And then this long sinuous snake winding back and forth is sort of a typical long read. They're just completely different technologies. And on the right are the error rates, per base error rates to scale again. So down here we have, I think, a 0.2% error.
Ben, you hit the mute button. You're muted.
Sorry about that. Did I hit it suddenly, or was I already into the slide when I hit it?
No, it's just been 2 or 3 seconds. It's fine.
Okay. So just on the error rate scale then, so the small bar is the sort of 0.2% error rate that we get with Illumina. And then on the right, I think was an 8 to 10% error rate to scale. It's just short read sequencing and long read sequencing are 2 different technologies. And they really work on different problems much of the time, or are effective on different problems. So let's think about this application of amplicon sequencing. And so one of the most common ways that this is used is to profile microbial communities. That's sort of the background that I came from. So it's an example that I like to use, although it's used many, many other places. And so in this application, what you have is you have a mixture of genomes, which are really what you care about. And what you do is you focus on a small barcode sequence from those genomes that you're going to amplify up. And so you can focus your sequencing effort on those barcodes. And we sort of intelligently choose that barcode so that even though we're sequencing just a small part of this genome, this barcode can tell us enough information to maybe say what genus or what species of bacteria or archaea or maybe even fungi it came from. Now in practice, what we're not using this typically to look at isolates. Isolates, we can just go sequence the whole genome. Where we're typically using this is on complex mixtures of different species and genera and strains. And so here in this little plot, I've expanded out what you get from one of these experiments. You get this huge stack of these barcode sequences. Some of them are from a black species, some are from a purple species, green, etc. And so one of the unique challenges of amplicon sequencing of mixtures is that we don't know and don't really have constraints on the mixture that we're measuring. And so we run into this problem where what if in my 12 millionth read from a sample, I get a brand new sequence, a brand new sequencing read I've never seen before. I don't know, I can't just say that's an error, right? So if I'm resequencing a human genome, boom, that's an error. I would have seen all diversity at the 2 alleles of this locus before. But that just might be a rare member of this community And I'd like to be able to know whether that's true or not. And what that means in effect is that accuracy is really critical for using amplicon sequencing on complex mixtures of communities. And so for this reason, amplicon sequencing using just the straight long read sequencing technologies with very high error rates has had a very limited impact in sort of a limited set of applications that it works on. And it's because the accuracy of those raw long read technologies was just too low in order to have any confidence about the new things you might see in the data, anything you don't see many times. So now I want to segue into what I'm going to call a new technology, and that is highly accurate long reads. And so long reads came about by fundamentally new sequencing technologies. You have the zero mode waveguides of PacBio. You have the nanopores of Oxford Nanopore. So highly accurate long reads are not coming from fundamentally new sequencing technologies. They're coming from a new strategy in which we are building consensus sequencing reads by getting multiple reads of a single long DNA molecule. And so that's how we're able to massively ramp up the accuracy while still potentially with innovation, maintaining our ability to create long reads. And so the sort of two main commercially available techniques for this is PacBio CCS or hi-fi sequencing, which is something that they talk quite a lot about now. And in that approach, so PacBio can sequence long reads. And so what they do is they take a longish read, maybe this is just a few kilobases, they circularize it, and then they use their long read sequencing technology to just read around that circle more and more times until you get a very highly accurate consensus sequence for that longish DNA sequence that you circularized. So this other technique that LoopGenomics has developed are called synthetic long reads because no true long read sequencing technology is used. This is based on short read sequencing, where the magic kind of comes in at the place where the library is prepared. So a single DNA molecule has a UMI or a barcode indicating its molecular origin that's intercalated throughout that molecule. And then when that molecule is broken up, we can create these short reads that can all be combined together. We know they came from a common molecule, so they can then be reassembled into that long DNA molecule. And the consensus of different, the consensus over those multiple reads can be used to create a very highly accurate long read sequencing read. And I'll admit, I'm not the expert on LoopGenomics chemistry, so if you have questions about that at the end, I'll be handing them off to the experts that are on this call as well. And hopefully I didn't say anything too wrong there. So if we go back to kind of comparing short reads, so the workhorse of the sequencing field, to what I'm calling HA long reads, highly accurate long reads, what we see is that highly accurate long reads don't go out to hundreds of kilobases. They kind of go out to tens of kilobases. But they also don't have a higher error rate. In fact, they have a lower error rate than standard Illumina short reads. In some cases, 1 to 2 orders of magnitude lower error rates. And so if we go back to that sort of a comparison plot I had before, here was our short read, then we have this, snake long read, and then our highly accurate long reads are in the middle. They're much longer than short reads, but they don't reach those tens of kilobases, or many tens of kilobases. But then on the error rates, they're also quite different. They are even lower error rates than those short reads had, and multiple orders of magnitude, 3, even 4 orders of magnitude lower per base error rates than just the raw long read sequencing technologies have. And so my argument is that these new technologies, which probably PacBio Hi-Fi was what really came onto the market and gained a lot of market share first. Now LoopGenomics is also able to offer a technology in the same class. It's effectively a third type of sequencing technology. It's a new sequencing technology. It's not long reads. It's not short reads. It's something with its own properties. And so I think that's very interesting, and it raises the question about in what applications and for what biological questions can this new technology be effective, and be applied to either do things better or to do new things that we couldn't do before. So I'm not going to focus on the full extent of that big question. I'm going to narrow in on the question of, do these highly accurate long reads really work for amplicon sequencing? So just focusing in on the potential technologies we're considering to focus on amplicon sequencing. Okay, so the first way that we try and ask that question, and I think it's a common one for people evaluating what ultimately we expect to use to measure complex mixtures, is we take what's called a mock community. So it's a defined mixture, in this case of 8 bacterial strains. And this is something that's provided by the company Zymo Research. So it's also defined up to certain thresholds, certain assurances about how accurate the different abundances of each bacteria are. And what we're going to do is we're just going to take that mock community standard, and we're going to try and combine it with the software that I've been involved in developing called DADA2. So you don't need to know about DADA2 too much. You just need to know this is a very widely used software for denoising, not denoting, amplicon sequencing data. And it was developed for short reads, where it's used quite widely now. But can we use this same sequencing technology, or this same software setup, with a mock community and LoopSeq highly accurate long reads? So we're just going to try it and see what happens. So to just start to give you some sort of practical pictures on what you see. So before this, we performed full-length 16S amplification from the Zymo mock community. And then those full-length amplicons were sequenced on the using the LoopSeq, just sort of standard lab service. And so the results look like this. What we're looking at is an output of the quality profile of the data that we got back. And so on the y-axis here are the quality scores assigned at each, on the x-axis, base position in the reads that we got back. There were 20,000 reads. You can see that the quality scores are almost universally very high, up until this position around 1,500. And that's expected because our 16S, full-length 16S gene is about 1,500 nucleotides long. And if you look at this cumulative read length distribution, you see that most of the reads are also extending out to about 1,500 nucleotides before they drop off to near nothing. So in all, this is a promising initial quality profile. Most of the reads are coming in at the read length we expect, most of the LoopSeq SLRs, and the assigned quality scores are quite high. DADA2 software package, we performed sort of the two standard pre-processing steps that we would perform at this point. So first, what we did was we used the function remove primers, so that both identifies the expected forward and reverse primers on each read. It reorients the reads if they need to, so that they all go from forward to reverse primer. And then it just cuts the reads. Anything outside the primers and the primers themselves are removed. And then the second pre-processing step is to simply filter on the length of the reads. So we have a strong expectation about how long the full-length 16S gene. So we filter things that are less than 1,000 bases and more than 1,700 or something like that. And we also filter based on expected errors. And so this can kind of be thought of as filtering on the average quality score, but in a slightly more sophisticated way. When we perform both of these steps on this data, we retained about 80% of the reads through our pre-processing, our quality filtering. That's great. And when we look at the quality profile afterwards, it looks very good. We have almost all high quality scores. Almost all the reads are centered around the expected length distribution. One thing I will note is that this fraction of LoopSeq reads that will pass the primer detection and filtering steps amongst data sets. And so I'll return to that a little bit later. But there's variability in the amounts of coverage each synthetic long read gets. And so that can lead to differences. Some data sets, this number could be lower than 80%. And so next, we just went on to perform denoising. And that happens in 2 steps in the DADA2 software package. We learn an error model. We learn how the quality scores and assigned nucleotides in LoopSeq data relate to the real ones. And then we use that error model to denoise the data, that is, identify all sequences that have errors in them and correct those errors. And so the result of all that, and so you don't need to understand every single step that just happened. What is important to understand is this is just with nothing added for LoopSeq at this point. This is just running essentially the standard DADA2 software on this Zymo community using this new LoopSeq sequencing technology, new to us. And so the result we got was a perfect, with an asterisk, reconstruction of the Zymo community. We detected all 8 bacterial strains. We detected every single unique 16S allele in all of those strains. So as a reminder, bacterial genomes tend to have more than one copy of the ribosomal operon. And so they can have more than one allele of the full-length 16S gene. And so what you're seeing in this bacillus facet here is that there was one allele that was present 4 times, a second allele that was present 2 times, and then 4 more alleles that are present 1 each. And together that makes 10 total copies of the bacillus 16S RNA gene complement. In addition to getting this complete ribotype, I mean, all alleles of the full-length 16S gene from each strain, there were no false positives in our output, and there were no residual errors on any of the denoise data after it had passed through the DADA2 denoising step. So this is very strong results, surprisingly strong to me at first, because we had done essentially no, we had made no changes from sort of on the software side from what we already had working for on Illumina, for example. And so that was the question that came to my mind, is why are things just working? And so in DADA2, we rely on an error model. So that is, there's this model for how a real DNA sequence goes through the sequencing process and turns into a sequencing read. And during that process, that real sequence S becomes a read R. Errors can be introduced into the string of DNA letters. So here's an explicit equation for that. That's not really important. But what's important to know about the error model is that the error model in DADA2 is allowed to depend on the kind of substitution. That is, an A to C error might happen more often than an A to T error, and that's okay. It's allowed to depend on the quality score. So an error at a site that was assigned quality score 40 will probably have less errors than at a site that was assigned quality score 10. And it's also allowed to vary from batch to batch. And so that is, each new batch of samples, the learn errors functionality in our software relearns the error model that you then use to denoise that data. But it's also important to know what the error model is not allowed to depend on. The error model does not take into account base position. So if the error model for the nucleotides at position 20 is really different from the error model for nucleotides in position 60, that would be a problem, and DADA2 would not be expected to work very well. It's not allowed, so strong nucleotide context is not accounted for in our model. So if you had errors where a motif like CCG was constantly converted to CCT, but that G doesn't go to T in other contexts, that again would be a problem for the DADA2 error model. And other kinds of gremlins that just don't match any reasonable quantitative model. And so why are things just working? Well, in this case, it's because the errors in LoopSeq data appear very well behaved. And so on this plot, we're showing in the same data, I'm just with the Zymo community data, from position 0 to position 1500, in these full-length 16S RNA gene reads. What we're plotting is the rate at which deletions on the bottom, insertions in the middle, and substitutions on the top happened at each of these positions. And then we're also kind of noting the quality scores at nucleotides that were in error. And so first of all, the first takeaway is that error rates are very low. So the rates of insertions and deletions are like below 10 to the minus 5, very low. Substitutions are by far the sort of dominant error mode here, but they are very well behaved. If you just scan sort of from left to right, there are no major peaks that wouldn't stand out from noise. The one feature which isn't just noise is that there is a slight elevation in substitution error rates at the beginnings and ends of LoopSeq reads. That's expected by the way the technology works, because it's building up a long read from short reads, and you'll get less short read coverage at the edges of the molecules. But that's okay, because the LoopSeq software is correctly assigning lower quality scores at those places. So given that the lower quality is correctly being assigned, that's fine. So these error rates are the error process in LoopSeq is very well behaved. And so that's why it works so seamlessly with sort of the DADA2 error modeling approach. So finally, the other strong assumption that DADA2 relies on is that in order to infer an ASV or a true sequence is in the sample, it relies on there being an error-free version of that true sequence somewhere in the data. So it will need at least one error-free read from that sequence. And so that puts limits. So for example, the very high, so if you're doing amplicon sequencing with just raw Oxford nanopore, there might be no error-free sequences in the data at all. And a DADA2-like approach simply can't work. Now in LoopSeq, because the underlying per-base error rates are so small, that means that the fraction of completely error-free reads remains high, substantial, all the way out through. So this circle here is for the full-length 16S data at 1,500 nucleotides. But we looked at other data sets. I think this was a fungal amplicon at about 2,400 nucleotides. And this was, I actually don't remember what the squares are, but they're about 5 kilobases. And so all the way up to 5 kilobases, you're getting nearly 75% of the reads themselves have no errors at all. And so that really empowers the sort of DADA2-like approach, and also kind of speaks to the quality of the raw data itself. You know, in many cases, this data may be usable without even trying to do denoising, because the fraction of error-free reads, at least if you're not pushing out into tens of kilobases, is so high. I will make a technical note. These figures here only consider reads in which both primers were detected. There probably is a higher error rate on reads coming out where the primers weren't detected, because some of that may be kind of misamplification of unexpected gene regions. So things worked very easily and straightforwardly because the properties of LoopSeq SLRs, synthetic long reads, matched up to the expectations of the DADA2 software package. And so you can get them working reasonably well together quite easily. But can we do even better by thinking about optimizing kind of the default DADA2 workflow for LoopSeq reads? So one thing to think about is that, I've made it small somehow. Okay. One thing to think about is that when we're talking about this 99.995% per base accuracy, we're really talking about point errors. And point errors are not the only type of error mode in sequencing, especially amplicon sequencing. And so another type of error that we might be potentially more worried about are what I would call structural errors. And so here in this little schematic, we have 3 different true sequences in our sample. blue, green, and orange. Our point error is a read we got out in which we read the wrong base at these two positions with the X's. Another type of error for which PCR is known is called a chimera. And that's where a read comes out in which the left-hand side of the read belongs to a different sample molecule than the right-hand side of the read. That often happens during the PCR process when you have incomplete extension at one PCR cycle, which is then used as a template in a next cycle. There's also a type of error mode we saw in LoopSeq data called an introgression. And in this case, there is a real sample molecule, the orange molecule, which has a relatively short segment in the middle that is introgressed in from another real sample molecule. So I believe we think we understand why that is. And this comes from the relatively rare cases in which multiple DNA molecules get the same barcode, UMI barcode. And so there can then be confusion when the assembler tries to assemble two different molecules into one synthetic long read. And this type of error can arise. So first, it's worth stating right now that both of those structural error modes are at very low frequencies. But one thing we wanted to look at is if we could use, so the DADA2 denoising algorithm doesn't remove structural errors. It handles like the point errors, but not the structural errors. So one of the questions we had was whether doing something as simple as optimizing our quality filtering could change the amount of structural errors we have. And so this slide's a little complicated. But on the x-axis, what we have is a filtering threshold. And so this is maximum expected errors. For those not familiar with that term, you can think of this also as kind of the average quality score. So where 0 would be a lower average quality score, 4 would be a higher average quality score. And then these lines are the fraction of all the reads that in the light blue line were chimeras when we had a very permissive quality score here on the right. So, 1.5%. And then when we had a very stringent quality score on the left, a very stringent quality filtering threshold on the left, the fraction of reads that were chimeras became much, much less. And what we see is that we can get rid of the vast majority of these structural errors, particularly the chimeras and introgressions, simply by imposing a reasonably, a slightly more stringent, sorry, I'm not sure why this keeps happening, but a slightly more stringent quality filtering threshold. The red line, if you'll note, is compared to the right side axis. And it shows you that this more stringent threshold of 0.5, we're still keeping nearly all the reads. We're keeping like 98% of all the reads, but we are very specifically removing those structural errors because they are assigned lower quality scores. So there is something in the construction of the LoopSeq reads and qualities that recognizes the confusion that might exist between different molecules and is giving lower quality scores to these structural errors. And so that's a really useful property. And it suggests that we can, in a very specific way, remove structural errors just by quality filtering. Now, the one thing that's a bit annoying is that, I'd love to say you should just use this max EE equals 5 threshold, 0.5 threshold. But this sort of optimal filtering threshold does seem to change from LoopSeq dataset to dataset. And that's related to the fact that there is some variation in the coverage per long read from dataset to dataset. But that's getting into some technical details. So that's structural errors. But the DADA2 denoising part is what deals with the per base errors. But this per base error rate is sort of lower than it was expected, or that the defaults for that algorithm were set for. And so given that this is about probably an 1.5 magnitude lower than the error rate we were expecting when we set the current defaults on our method in the software package. And so what we determined is that we can increase the sensitivity of this denoising step with these two parameters. One of them is called detect singletons equals true. So this allows you to detect true singleton sequences in your data. And the other is this omega A parameter, which I'm not going to get into the details of what that is, but we recommend this more sensitive value for that parameter. And just to show that this does really work, and now moving to a different data set, this is a real human fecal data set. So again, we're doing full-length 16S sequencing on a human fecal sample. And what we're evaluating at the end is we performed our denoising with DADA2. At the end of that, we get a set of sequences that were split from one another during the denoising. So I have sequence X and I have sequence X prime. They were split apart, and I can look at the differences between them. So X and X prime were split apart because they differed, an AC at position 100, a G to A at position 345, and a to a T at position 1012, something like that. And what I'm going to do is I'm going to record the number of differences in all these splits that occurred at conserved regions of the 16S RNA gene. So that means they're conserved evolutionarily, or more evolutionary variable regions of the 16S RNA gene. And so first, the sequencing technology does not know about the conserved and variable regions of the 16S RNA gene. They are conserved and variable because of the biological function it performs, and the fact that if you make changes in conserved regions, you're likely to hurt or even kill the offspring. The sequencing machine knows nothing about that. So if sequencing machine errors were being propagated forth into our denoised data by this high sensitivity inference, we should see a lot of points on this dashed red line. So that's what you would get if you just randomly put errors wherever on the sequence. Instead, what we see is that essentially all of our splits here fall on this gray line, and that gray line is showing that there is a highly enriched number of differences between related sequences in the evolutionary variable parts of the 16S RNA gene, not in the conserved regions of the 16S RNA gene. And so, one of the difficulties in evaluating in complex mixtures is that it's hard to have a gold standard oracle truth in complex mixtures. But this is quite a useful way to evaluate that this pattern strongly suggests that we are discriminating differences that very strongly match sort of the biological expectations of where those differences would be, and don't match at all what we would expect if sequencing errors were an important part of driving the denoise diversity. And so one other example on these full-length 16S from human fecal samples, which I kind of like. This is in some sense a boring slide, because what we did was we have 3 different human fecal samples, and we performed full-length 16S sequencing using LoopSeq. We also used PacBio CCS, 2 different chemistries, and then we used DADA2 to reconstruct the composition of those samples at the ASV level. And then we just plot them on this ordination. So the closer an X is, the more similar a community composition you got. And what you see is that the technology is completely irrelevant. Every one of these highly accurate long-read amplicon sequencing technologies is getting the same description of each sample. And so that really, I think, raises the confidence in all of these technologies. I mean, if they're finding the same answer, then there aren't technologically relevant gremlins hiding in there. They're finding the same thing because that's probably the right thing. Okay, so what I've tried to convince you to this point is that there is this effectively new technology of highly accurate long read sequencing, that it can and does work for amplicon sequencing, that it actually marries very well with the DADA2 method, which in theory can kind of get the maximum out of this data because it will resolve this data down to the single nucleotide level, rather than imposing sort of a higher order clustering threshold that's would otherwise commonly be done. And one of the questions I have, and I don't have the answers to this, but a thought on this is where this fits into a large area like microbiome profiling. And so there are sort of multiple tools that now are available. So here the idea is that I have some host-associated microbiome sample, and I want to get a profile of the community. So there are sort of, right now, sort of two dominant technologies here. So one is short-read amplicon sequencing. So like short-read 16S is the classic version. That's the cheapest. It has very limited ability to resolve. Usually you're getting down to saying what the genus is, and that's about it. Alternatively, as sequencing costs have gone down and tooling has gotten better, shotgun sequencing has become much more desirable as a method to do microbiome profiling. And it's probably preferred if you want higher resolution than short-read amplicon sequencing. But there's this additional factor about the amount of what I'm calling host contamination, but the amount of DNA that's in a sample that isn't coming from the microbial community you actually want to assay. And so as host contamination increases, and so different types of, in the human microbiome, different types of microbiome samples have very different fractions of host contamination, then the amplicon methods become more desirable because the amplicon process, that initial PCR targeting process, pulls out the bacterial or microbial fraction and gets around what could be a vast majority of human DNA that otherwise would yield wasted sequencing. And so where I kind of see highly accurate long-read amplicon sequencing fitting in here is that it will become very useful, I think, for being applied to samples in which there is moderate to high amounts of host contamination, in which there is a desired resolution of the microbial membership that's above the genus level. So if you want species level resolution or even above species level resolution, then this technology, I think, can be a good solution in that space. But certainly all three of these technologies will continue to have particular applications where they're sort of the right choice. And to give you one area that we're interested in, and also point out that there really is open space and really being able to leverage what is possible with these highly accurate long-read amplicon sequencing technologies. We talk about species level profiling, and that's certainly something that a lot of people want. But the accuracy of the data and the length of the amplicons we can now generate will allow us to go below the species level and identify relevant subspecies variation in at least some cases. And so one example case of this, and something that we're working further on, and particularly in the E. coli and Salmonella genera, is in the Zymomock community, when I'm able to find this complete allelic fingerprint of the 16S gene, that actually allowed us to identify that this was E. coli 0157H7, which is one of the most important foodborne pathogens and a bug of great commercial relevance because of its importance to the food supply and the amount of time we spend projecting the food supply from it. And so that's subspecies variation that we can identify now using these existing technologies. Now the methods and databases to do that more generally aren't there yet, but I think that they will come over time. So another example, what's another area of genetics and genomics where we often look at a population embedded largely in other DNA? Well, that's anything to do with viruses, right? So when we're doing viral genetics, viral genomics, typically we're doing some kind of PCR step in order to amplify up that small amount of viral DNA that then we want to sequence and understand. So for example, this is one of the common protocols, at least as of about 3 or 4 months ago, to do SARS-CoV-2 genomics, and it involves tiling, I think, about 96 amplicons because we're doing short-read amplicon sequencing here. So we need 96 primer sets, 96 amplicons. We get this very choppy coverage distribution over the viral genome. It's not that this doesn't work, but there's certainly the possibility that highly accurate long reads could reduce how we could reduce the complexity of this type of viral genomic sequencing greatly. And in viruses in which there's sufficient diversity, it could start to provide long-range linkage information that could link, mutations at different parts of a gene, or even in one gene to another gene, when we have multiple variants of the virus co-circulating in a population we're studying. So I think there are a lot of other potential applications. Again, as this technology is really quite new, we're still learning about it. Certainly metabarcoding, I think, especially in eukaryotes, which it's much harder to do to go the shotgun metagenomics route. Tumor sequencing, I mean, certainly there's been, it's really changed our understanding of the heterogeneity of solid tumors and how important that is to their progression. Looking at the immune repertoire, functional genes, doing high-ploidy genetics. I don't know if any of you guys have worked in high-ploidy plant genomes, but that can be a real nightmare to phase, the allelic complements on those things. And so this type of sequencing can really help with that. So I'll note, we continue to support long-read amplicon sequencing. To be fair, we support highly accurate long-read amplicon sequencing. So sort of PacBio CCS and LoopSeq right now. And you can find out more information about that with the, at the web page and help forms associated with our software. And so finally, just the takeaways here. So LoopSeq sequencing is very, very accurate. It's also well behaved in a way that allows DADA2 and LoopSeq to just work together. So we do suggest using higher sensitivity settings, both to take full advantage of the data and an acknowledgement that as we have longer and longer amplicons, you have a higher and higher fraction of the data that's at relatively low frequencies. In practice, for people putting DADA2 and LoopSeq together, the pre-processing steps are where you will make the most important choices. I think we just got a report this week that something about the way our remove primers method works was completely losing a clade of cyanobacteria. So it's in those pre-processing steps where I think improvements in default choices and where improvements in parameters that a user might set themselves will make the biggest impacts. I will say we're still learning about LoopSeq in the wild. We have looked at LoopSeq data in a couple different types of data sets. All these data sets were pretty small, kind of on the evaluation scale. So certainly we're interested in different experiences, and so we can start learning about LoopSeq in the wild and help real people. I also know there is variability in data set quality expected. That's part of learning about LoopSeq in the wild. I don't know how much, but some data sets that we looked at had higher average quality than others, and that's expected with any sequencing technology, but it's something to keep in mind. And you can also read in a lot more depth. So we have this preprint up, which should be published if I could get, it's my procrastination, which is not reason it's not published yet, but it will be pretty soon. And alongside this manuscript is these complete workflows for how we processed and analyzed the LoopSeq data in that manuscript. And so these are all completely reproducible on your own if you'd like, and can be usable as workflows for your own data as well. Okay, so just want to thank everybody, people working with me on the bioinformatics applications technology, both at Loop and PacBio funding, USDA and NIH. I also just wanted to specifically call out my postdoc, Dmitry Grinovich, who is going to be working on that subspecies profiling, which I'm kind of excited about, very specifically related to, can we tell a pathogenic E. coli from a non-pathogenic E. coli, or the like. Okay, so thanks for listening. So I think that we're doing questions now, and I'm sorry, I did go about 10 minutes later than I meant to, but I can stick around, and I think that Tony perhaps is going to read questions, though.
Yeah. Hey, Ben. Thanks for the presentation. Really exciting data and very interesting. I think I can tell just by the amount of questions and the depth of the questions that everyone really enjoyed your presentation. I will read out, at least I'll try the majority of the questions. And if we don't get to all the questions, we will be sure to follow up with the people that wrote questions and answer them over email if we hit a time constraint. So I'll try to group the questions into kind of different categories. You know, if Ben, if you feel that you want to, you know, someone from the Loop team to, I guess, me to jump in and answer questions that are more related to the chemistry side, just let me know.
Okay.
Okay. Okay. Questions are still again. So just so you know, a lot of the remarks are that was a great talk. So thanks again for that. Okay. So let's start. So first question is, are introgressions Illumina errors or PCR errors?
Yeah, so I think Tuval might know this best. From what I understand, they are essentially assembly errors. So they happen when you're assembling the long read out of the short reads. And I think that it's, as I hinted at, if you accidentally give the same UMI to two initial long template molecules, then the assembler will try and put together those two into one read. And the result of that can be it spits out an introgression. What's nice is that the introgress region tends to get low quality scores because the assembler kind of knows it's confused there. And so that's why this quality filtering approach works really well to remove them.
Yeah, that makes sense. I think kind of, so it's not really an Illumina error or a PCR error. It's more of a sample prep error that can be relatively easily removed, as Ben mentioned, through quality filtering. Okay. Next question is, I guess, what do you sequence here, the entire genome or just the 16S read or just the 16S gene in the work that you've analyzed, Ben?
So the majority of the data I showed in this talk was from full-length 16S sequencing, but we've also looked at data that came from just amplifying random bits of genomes, so there's spots all over the genome, and then also from amplifying other marker genes, so the ITS or greater ITS 23S or whatever it is region in fungi. But I am not looking here at like shotgun metagenomics with LoopSeq, which is, it's not that you can't do that, it's just the focus here was entirely on amplicon sequencing.
Got it. Thanks. Another question here related to length is 10 or slightly more KB the maximum length limited? Basically, where are the length limitations coming from? So, I mean, I think I can answer that question. It's a chemistry question. So basically, the length limitations are coming from the length limitations of long-range PCR. So the technology is essentially limited to the length limitations of long-range PCR. So if you can make a 5 KB amplicon, we can sequence it. If you can make a 10 KB amplicon, then LoopSeq can sequence it. So basically, the length limitations come from the limitations of PCR. Right. Next question, interesting question, is that there have been recent publication that tries to couple ONT with PacBio with UMIs. And by doing that, trying to error correct the reads. So basically trying to sequence, you know, ONT or PacBio reads with UMIs at, say, 25x coverage, so that each molecule is sequenced multiple times and trying to error correct using that. So basically, how does DADA2 perform better to profile bacteria, in that case, in the complex microbial community? So Ben, I'll let you answer that question from a computational point of view, and then I think I will add a few points from the chemistry point of view.
Yeah, so I'm also very enthusiastic about that. So I think it just got finally published last week, although it had been out in a preprint for over a year before that. It could work very well with DADA2. We haven't tested on that type of data, and so I just can't say for sure. But I think that's, although it's not a commercial product yet, I think that's another entry in this category of highly accurate long read sequencing technologies. So I mean, I think it's exciting.
Yeah, definitely. Three comments I think that I'll make from the chemistry point of view about applying UMIs to long reads. There are three challenges to doing that for basically the PacBio and the Oxford Nanopore approach. One is that really this approach can only be used for high biomass samples, because the UMI attachment step cannot be done in a PCR fashion. It has to be assigned directly to molecules. So, and that only works in high biomass samples. So samples like skin samples or any really microbiome sample that has low concentrations of DNA would be the UMI assignment step would basically not work. It requires a relatively high DNA concentration. Another challenge that we've seen with that is that the error rates will vary greatly because some molecules will have 10x coverage, some will have 1x coverage, and some will have 200x coverage. And it's very hard to control that. It basically depends on the initial concentration of the microbiome. If you're sequencing a low biomass sample, let's say that you're going to sequence all of your samples with 10,000 PacBio reads, whether you originally tagged 1,000 molecules or 10,000 molecules will make a big difference on the final fold coverage of your sample. So you might end up with one sample with 1x coverage, which will give you no error correction, and with another sample with 10x coverage, which will give you substantial error correction. That's the second challenge with that. And then the third challenge, of course, is that, well, maybe not a challenge, but just it's a matter of cost. If you have to sequence each molecule 10 or 20 times to reduce the error rate, it basically increases your sequencing cost tenfold. So if you're sequencing each molecule 10 times, that means that you need 10 reads for each read that you're actually going to get. So those are the kind of the three comments about the challenges of trying to integrate UMIs into true long read sequencing in the context of 16S. Next question for you, Ben, is, are these data usable with standard taxonomy classifiers?
They're usable. You're just not getting your full money's worth, probably. I mean, so that's one of the places where new work is needed. So we've made some small steps towards, providing a species level Silva database that works with our standard classifier, and that works to do species level assignments pretty well. But we don't have great tools, at least not ones that I'm aware of, for doing species and below level assignment from like full-length 16S or 16S plus, just because it hasn't really been required until recently. I do think there might be some pretty decent commercial tools out there. So I know a company called Shoreline Biome has been working on this problem probably longer than many people in the academic field. There was another company called Smart Gene that got in touch with me and kind of compared their assignments from this type of data to ours, and I had to admit theirs were better. So better solutions are coming, but at least on the academic, open source area, improvements definitely can be made.
Great, thanks. The next set of questions have to do with thinking about throughput and cost considerations. Very interesting talk. Would be good to know about the trade-offs. What is the cost per sequence library? Another question related is, how does throughput compare to short read and long read sequencing? How many samples can be run together using LoopSeq technology? And what is the typical library size per sample? Ben, I don't know if you have any thoughts about that. I can kind of go after you.
Yeah, so I think that the economics on this have been changing quite a bit so recently that as someone who doesn't actually, I run a dry lab and I'm not constantly sequencing, I feel uncomfortable making strong economic statements. I will say that the costs of doing this type of sequencing has gone down with things like the Sequel II instrument from PacBio and from the ability to do LoopSeq style where everything's run in an Illumina sequencer. But I'm sure Tuval probably has more to say.
Thanks, Ben. So just a few kind of data points. So in terms of throughput, and coverage. LoopSeq aims for roughly a 20 to 30x coverage for each molecule. So that means that every barcode, every long read, every 16S long read requires roughly 20x coverage of short reads. So that's the amount of additional sequencing that you need to do. In terms of how many samples can you actually fit on a run? So I can give you a typical example. So on a high-seq lane, a high-seq 4000 lane that has 800 milli reads, you can fit roughly 200 samples, and that's assuming 10,000 long reads per sample. If you want to translate that to sequencing costs, that's roughly $5 to $6 per sample in terms of sequencing costs. And then, of course, if you can run that on a NovaSeq, then costs are even maybe 30 to 40% lower than that. Yeah. So, and then another thing to mention here is that typically in the microbiome space, people used to sequence the 16S gene with the MiSeq instruments from Illumina. The reason they were doing that is because the MiSeq is the only instrument, the only Illumina instrument that can give you paired end 250 or paired end 300 reads, and you need that in order to cover 2 variable regions and not one. But the problem is that the MiSeq is by far the most expensive Illumina instrument in terms of cost per base. Well, one of the major advancements with LoopSeq is that you no longer need that MiSeq 300 base pair reads. You can use just a standard paired end 150 Illumina run on any instrument, a NextSeq, a MiniSeq, or a HiSeq, or a NovaSeq. And those reads are roughly 10-fold cheaper per base pair compared to the MiSeq. So you do need to do extra sequencing. You need more data for LoopSeq, roughly typically 3 to 4 million short reads per sample, but those reads are much cheaper than the typical MiSeq reads. So they those two factors kind of cancel out. Another question is, does LoopSeq have any plan to cooperate with Illumina to make highly accurate long reads sequencing more accessible? So the short answer to that is that it's already very accessible. You can buy our kits or use our service through our website, and you can run our kits on any Illumina sequencer. Next question is, how does LoopSeq handle quality score for each nucleotide when merging all of the short reads? Ben, do you want me to answer that?
Yeah, so I'll say one thing. So yeah. I don't know, really. I mean, I know it relates to kind of the assembly and how consistent it is. But one of the things, the DADA2 side, DADA2 does not assume that the quality score meets its nominal definition. What DADA2 sees is that there is a consistent relationship in LoopSeq data between the quality scores they give and the actual error rates. So in some sense, whatever they're doing, it's producing a consistent quality signal, which is good.
Got it. Thanks, Ben. I can give a little bit more detail about how we're actually calculating the Q score. Basically, we have an error model, which is basically depends on 3 parameters. One is the coverage. So we calculate a Q score for each position in the long read. So what we end up with are Q scores, and we generate FASTQ files. So the exact same standard FASTQ file of the Illumina format is generated only with long reads instead of short reads. So each base pair in the long read has a Q score, and the Q score at each position in the long read is calculated based using our error model that takes into consideration 3 factors. One is the amount of short read coverage at that position. So whether we have 5, 10, 50 short reads that cover that position, that's one factor that we take into consideration. The more short reads we have, the higher the Q score. The second parameter that we take into consideration is the actual Q score of those short reads. And then the third parameter that we take into consideration is the level of consensus between all those short reads at a given position. The higher the consensus, the higher the Q score. So those 3 parameters are what goes into our error model, which that Ben analyzed and found that had a good correlation to the actual errors in the sequence. Right. Next question. Will bias caused by PCR cycle affect accurate quantification? And will it require, and does long-range PCR require a high-fidelity enzyme? So Ben, do you want to address the first question? Do you see any bias caused in PCR in terms of quantifying microbiome samples?
Well, so long reads are not going to get rid of the problem of bias in sequencing the microbiome. And bias isn't just from PCR, it's from DNA extraction, it's from, sample storage, et cetera. That's why when I said we got a perfect reconstruction, I put an asterisk on it, because it's not perfect at the level of getting the abundances exactly right. We didn't try and rigorously evaluate the contribution of PCR in like the, in like LoopSeq kits to additional bias here. The one data point that I could point to is that both LoopSeq and PacBio Folding 16S were getting very concordant results, which includes the measured relative abundances. Although to be fair, they were using basically the same primer sets as well. So I'll just stop there. I'm kind of rambling.
Got it. And in terms of the ability to quantify synthetic correctly quantify the relative abundance of synthetic communities, were those were the abundances that you saw for LoopSeq and PacBio, did they correlate with the expected abundances of the synthetic community?
So we did not try and evaluate that. We were using, this is maybe a technical point, but an important one. We were using their DNA standard, where the abundances are quantified at the level of DNA. Whether or not you can recapitulate the DNA abundances, that doesn't mean you would recapitulate the abundances in a real sample, because real samples come in cells, and DNA extraction can be the dominant, or is often the dominant bias factor.
Got it. Thank you. Can the same technology be applied to shotgun sequencing?
Sure.
Yeah, definitely the answer is yes. And we have, and also some of the data that Ben presented today was made through a shotgun sequencing protocol. Are UMIs assigned through droplet technology?
That's you to all.
Yeah, so I mean, the short answer is no. They are not applied through droplet technology. That's one of the major advancements in LoopSeq technology over previous synthetic long-read technologies. We've completely circumvented the need for any hardware whatsoever in terms of sample prep. All you really need is a PCR machine, so there's no microfluidics or any type of microfluidics or droplet technology. They're not needed. Right. One question, which I think you addressed during your call, but given LoopSeq is already accurate, how much can DADA2 further denoise or enhance the ASV endpoints?
Yeah, so I mean, I think that in some applications, if you don't care about the very rare fraction of the data, for example, you could do something much cruder and get very similar results. I think that what we were looking at in terms of the quality filtering to get rid of structural errors can be important. But I believe if you do something really simple, like I'm going to impose a threshold that I believe everything I see 3 or more times, that might, I give up on everything that I would see twice or once, but given the accuracy of LoopSeq, that might actually work. So, at some point, a 100% accuracy technology doesn't need denoising at all anymore. So I think that's a relevant consideration. And I think DADA2 has some value added in being able to get down to the rarest fraction of the data. But if the data quality can be good enough to just skip that step for some applications.
Thank you. Can this technology be applied to IPS or ATNS to identify non-bacterial or archaeal species such as fungi?
Yep, if you can amplify it, you can use this.
Yeah, that's basically the answer. So you can basically do long-read sequencing for any amplicon that you make. We also have, LoopSeq also has a dedicated ATNS ITS amplicon, roughly 2.5 kb amplicon that is already packaged in the form of a kit. So that's a kit specifically designed for high-resolution identification of fungi. How about the resolution of long sequences for identifying intraspecific or intraspecies, I guess, is what they mean, variations? How is DADA2 dealing with that?
So DADA2 itself, the core algorithm, its target endpoint is all the real sequences in your sample with no errors. The downstream applications of that, we do provide in our package taxonomy assignment for microbes because it's such a common second application. But we have not tried to build anything, specifically for interpreting intraspecific variation. Certainly I've seen people doing that even with short reads, trying to understand kind of population sizes, maybe by looking at intraspecific diversity within sort of like different protist species, but it's not something that we directly support.
Got it, thank you. Next question. Does DADA2 handle full-length 16S analysis, and is its analysis of the full-length 16S gene the reason why you can capture subspecies variation?
Yeah, so it'll handle it down through assigning to the species level. At the point in which that point that I showed us identifying 0157H7, that was basically done by hand. So we identified, and we've done this in real samples as well. When we identify a strain of interest, we can, but this is at this point it's by hand, we can extract its full ribosomal complement and then use a couple different approaches to try and narrow down that strain to some clade or some subspecies clade. That's not automated right now. What I'm hoping is that we'll have a publication in the next year or so where we'll have kind of an automated solution for certain bacteria that we're working on, but that's work in progress.
Got it, thanks. And then last question, can we use our own primers, or are there primers hardwired to each kit? I guess I can answer that question. So the 16S kit has, so to speak, hardwired kit primers in it, and the sequences of those primers are published and they're on our website. Having said that, if you want to use your own primers, we also have a general amplicon kit where you can just make your own amplicon and of any kind, not just 16S, and sequence it as just a general purpose amplicon kit. Ben, one last question for you. Follow-up question on whether the sequences were 16S or entire genome. If it's 16S only that was sequenced, why was the length of the sequence shown in the plot more than 1,500 base pairs in the graph that showed that the accuracy was reduced after 1,500 base pairs?
I don't know. I mean, at that point, I think that the length includes the primers themselves as well, and may include some adapter outside of them. I don't remember. Once you cut down to between the primers, then they pretty much all fall below 1,500 at that point.
Got it. Okay, thanks, Ben. I think those are the major questions. If we missed any questions, again, if we missed any one of your questions, we'll follow up over email and try to answer your questions for our tech support team. Ben, thanks again for the presentation. I can tell you that there are a lot of comments about how the presentation was great and really interesting. So thanks again for that. So this will conclude our Q&A session, and I will pass the torch here to Tony, who will say some concluding remarks and some housekeeping items.
Thanks.
Thank you.